Okay, and we are back again with our 5th annual Big Give. I am always amazed about the amount of people who are so invested in Do15Q syndrome and have grown with our organization, such as Dr. Vidya Saravanapandian. I remember our very first Big Give, it was 2018, and I believe we interviewed with you and Dr. Juste about EEG biomarkers and Do15Q syndrome, and it's just continued to grow into your most recent was the EEG in sleep. So welcome. Congratulations on being officially a doctor, Sarah or Vidya Saravanapandian. And tell us what you're up to.
Yeah, so I graduated last year and I'm now a postdoctoral research fellow in UCLA. So yeah, I decided to continue my work here, my research here in UCLA. It's such a great community. And most importantly, I decided to continue my work in Duke15Q syndrome, because I was just so excited about the work. That's all my PhD was about. And so, yeah, now as a postdoctoral research fellow, I'm really trying to expand what I studied as a graduate student, but also try to, make some progress into next steps that will really help us move forward with clinical trials in the future, hopefully. And I'm excited to tell you guys about that.
I think it's interesting that, as Neka pointed out, I think it shows the value of our research dollars at work, from looking at, years ago, investing in you as a fellow, and then now moving through the process. It's just, it just kind of makes me go, wow, we need to be doing more of this. Like, I kept thinking we got to do this more, because we want to raise up that next generation of physicians, and doctors and researchers. And I think this is such a fantastic investment. What has kept you interested in Duke 15 through this many years? What's been like, has there been one driver for you?
Yeah, so I was born and raised in India. I grew up in India, and I grew up in a community where there's still, there's not much awareness for neurodevelopmental disorders in general, and there's such a high stigma towards autism as well as other neurodevelopmental disorders. And I've always been interested in sort of understanding the biology behind such disorders. Not only to improve treatment, but also to increase education about these disorders in the community as well. And so when I joined Dr. Shefali Juste's lab, I was really excited about the work that she was doing at the time, which is really trying to identify brain-based biomarkers in children with neurodevelopmental disorders, particularly Duke15Q syndrome. And I had some background working on deletions of GABA receptor genes and syndromes associated with that in the past as an RA when I was working at Stanford University. So when I came to UCLA, I was like, oh my God, this is perfect. This is what I really wanted to work on. And then when I started working on the project, I got this amazing opportunity to meet with the families, which I think is very rare, because I was a basic scientist, right? Like, as a basic neuroscientist doing bench work, being able to make that connection with families is really important. And Dr. Shefali she gave me the opportunity to actually shadow her at the clinic, at the genetics clinic every month. And also part of my research was to be able to collect EEG data from our children with Duke15Q. So I was able to meet them in the research lab, and also understand not just at the data analysis level, but, at the genetic level, what is happening, and also interacting with them and making that connection was extremely motivating. And that's what kept me going. And I think when I was deciding, okay, what am I going to do after my PhD? And after all of this amazing work, then I decided, okay, I just want to really continue working on this syndrome and really be able to move on and make some progress that'll actually make a difference someday. So yeah, that's what keeps me going. And I'm really excited that, as a PhD student, I was able to work on clinical data, work with, again, as Neka mentioned, I worked on this sleep physiology project where we were able to connect with families and gather a lot of sleep EEG data. And analyzing that, I found this profound abnormality in sleep physiology that is occurring in Duke15Q. Really including, abnormal deep sleep state, which, as you can imagine, is so important. Sleep in general is so fundamental. It's important for our normal cognitive function, but having these sort of disrupted sleep physiology, including not being able to achieve that deep sleep state, it really impacts. For everyone, I think not having a good sleep has, such profound impact, but also when you already have behavioral issues and when you have high levels of anxiety and other things going on, I think when you don't get good sleep, then that really, exacerbates your vulnerability and make things worse. So so after having found those, now as a postdoctoral research fellow, I'm actually doing taking a translational approach to understand, okay, we know that with the clinical population, this is what is happening, but using advanced technology as well as utilizing DUP15Q mouse models, can we now be able to recapitulate these things? And if we are able to understand what's happening at the biological level, then we would be able to, intervene and do manipulations in mouse models where we have these genetic mutations that's occurring in patients. And then just characterize the sleep physiology in mouse models to be able to manipulate and maybe, figure out a way to treat this sort of abnormal EEG signatures that we are seeing in patients, so that we can see changes overall in development as well as cognition.
So just to circle back for parents who are tuning in and maybe they didn't hear your presentation for a Big Give last year, which I will put in the comments, that while you were looking at these sleep EEGs, you did see consistently that the kids, was it the REM sleep that they weren't getting into? What sleep stage that they just couldn't get into? I can't remember.
So yeah, it's the non-REM sleep, so the non-REM sleep. So which is one of the stages. So within non-REM sleep, we have distinct stages where we have sleep spindles, which are basically like high bursts of oscillations that come and go. So they are called sleep spindles. So that happens in stage 2 sleep. And then there's stage 3 sleep, which is a deep sleep state. And the stage 2 and stage 3 combined is the non-REM sleep. So both are affected actually. And both spindles, as well as these this really slow oscillations that occur in deep sleep state, they are both really critical for long-term memory consolidation and, brain plasticity. And so when we are not achieving that, during our daily sleep, then that has significant impact. And also, through years of research, we've learned that there needs to be a temporal coordination that needs to happen between the sleep spindles as well as the slow oscillations. And we think that that's also not happening because of all these disruptions in these different sleep stages. And also, if you remember from our past meetings and presentations from Dr. Jeste and that we have given in the past, like there are also these higher frequency beta oscillations that also occur, which are these, clear signatures that we see in DUP15Q EEG. We also see that in sleep. And so, everything all taken together, there's these abnormalities that happen in sleep that could impact highly.
We were talking with Dr. Ron Thieber earlier today, and we were talking about seizure control and things like that. And one of the things he had said was, when kids aren't getting good sleep, it is exacerbating their seizures. And sometimes, no amount of medication at this point is going to help control these sort of seizures when they're not getting this sleep. And I remember having this conversation. It kind of comes full circle for me. If you're identifying that our kids aren't sleeping and you can figure out how to measure that, which is what you say you're doing, there's hope that we could help our kids sleep better, thus lessening all of these symptoms of seizures and, the autistic behaviors that are causing our kids not to live their best life. So just hearing all of that, we didn't prep anybody to have these conversations beforehand. So listening to it,
same thing, Nika.
It's just like, man, it's wonderful to see everything really come together in the work that you're doing. So now that we've got the EEG biomarkers, about the sleep, you want to go in translational research. What are you excited about next?
So for my current work, so I'm actually recording sleep EEG from our DUP15Q mouse models. So just like how we recorded from patients, so we are able to look at the scalp level, so record sleep, and just look at all of these signatures that I just mentioned. So quantify sleep spindles, looking at slow oscillations, and we have actually collected quite some some data, and we are looking at the data now. And what is exciting is we do see some differences in the slow oscillations, or the slower oscillations, or the oscillations that we see is abnormal in patients, is also seem to be affected in mouse models as well. I mean, we still have a long way to go. Like, it's just a preliminary data that we are looking at, but it's I think it's exciting. It's going in the right direction, and we want to continue. And we are also able to use technology that we have. So I'm a postdoctoral fellow in Dr. Payman Golshani's lab, and we have these miniaturized microscopes and other tools that we can actually use to be able to record from multiple brain regions in mice when they are sleeping. And so we can also look at that sort of temporal coordination, or, you know, the timely synchrony that's supposed to happen during sleep. We can also quantify that, which is really exciting. So yeah, so that's what we're doing now.
It's amazing to me the way that technology has advanced, where you can, like, I think it was Ben Philpott and Matt Judson were talking about putting like a little EEG thing on the mice and be able to like have them walk around with it and measure all of these things, and be able to put this on the mice and see all parts of their brains while they're sleeping. Like, it's just amazing how advanced medicine is. And it makes me hopeful for the future on how, with people like you working in medicine and working for our kids in research, how more advanced it'll be in the next 5 years. Like our 10th annual Big Give, what are we going to be talking about?
That would be fascinating to think about.
Yeah.
Yeah. No, it is exciting. And sleep is, you know, the sleep impairments are common among a lot of neurodevelopmental disorders. And I think it's a really important issue that we want to be able to understand and help with treatment. So I'm really excited about that. And I want to also, again, thank the Do15Q Alliance, because this all wouldn't have started if you guys didn't give me the fellowship. You know, that was the start of this work, really, in 2017 when I got the fellowship. And I was able to meet families at our conference and actually collect data. Over 3 days, we collected EEG from almost 40 families, I remember. And so, yeah, that was amazing. And that sort of was the beginning of this whole sleep work. And it's still going on, continuing.
Well, sleep and then just the EEG biomarkers in general. Are you still collecting EEGs? Is that something that families could still submit for research, either to you or Dr. Jesse?
Yeah, definitely. So families can send me, I think we have, so in the past, Vanessa was able to help me gather EEG through the Alliance and then send me the data sets. And then I think some of the families were able to send it to us directly, also to me and Dr. Juste as part of the study. And yeah, so there are two things that I'm working on now. So one is the mouse model work that I just mentioned, but also the other arm is just to really expand our clinical study as much as possible. And in the future, I'm also hoping to expand this to other disorders, really, because once we sort of standardize all of the, data analysis and everything, we should be looking at, related disorders closely so that we are able to even understand and learn from other disorders, because there's definitely a lot of overlap with the behavioral phenotypes that we see. So we know that at the molecular level, at the physiological level, there should be an overlap as well. So when families have more EEG, sleep EEG that they would like to share if they haven't done in the past, we would love to have the data if that was a possibility. And I'm happy to, you can share my email, and I'm happy to connect with them to, let them know what the process is and what else I would need. Thank you for that.
Vidya, one thing we were talking about today is, we've been talking about fundraising for clinics and research and Ladder, of course, our database, our medical database. And one thing I think that's so poignant to me in this conversation is that the research dollars that we invested in you has now come full circle, as we've talked about. So that's the type of money we're raising today, and be able to invest in not just the future of therapies, but even the long-term future of therapy. Because I mean, looking at how you have been able to go from a student we invested in to now you're like, I fell in love with this community And I feel that too, because I'm the same way. I've fallen in love with this community and I'm like, what can I do that's more? And it seems like you're the second person we've talked to today who had that same type of experience. So I want to tell people watching that that is what we're raising dollars for, is investing not just in therapies, but the future of therapy and the future of research. But I also thought something you said that was so important is getting those EEGs. And we have said this, I know a lot today, but I know people may listen to this in segments. And I'd be remiss not to mention that this, what you're talking about, is exactly why our medical database is so important. Our ladder database, putting those EEGs in so that researchers like you can pull not just 3, not just 4, but as many as people will put in there. So you have this plethora of research. Give us our last little pitch here on what does that mean to you when you can go and request data and just have a slew of data, if you will, to be able to go in and research.
Yeah, no, thank you for bringing that up. So yeah, that's a really important aspect of all of this work. It's fundamental to our work, right? Like being able to share data and being able to access the data. Because I know that there are a lot of families who have collected EEGs in the past and other behavioral assessment and everything. And to be able to get that data has been so important. And in the past, as many of you know at the Alliance, that has been the most time consuming part also. And then I totally understand, on the family side, it is a lot of effort. And I'm very thankful that, last year, I was able to connect with a lot of families during the pandemic. I was able to connect via Zoom, talk about the project with families, and they were able to send me directly their EEG discs. And I'm so appreciative of that because, it takes a lot of effort to be able to do that when you have so much at home to take care of, along with, during the pandemic, you're working from home, you're taking care of your DUP15Q child and everything. And so we're very thankful for that. And so a common database like the ladder that you are developing is so important so that it's easily accessible for both families as well as the researchers. And I know that it doesn't take much of time to be able to upload the data from home. And for us, if you just give us the access, then yeah, that really accelerates our research so much.
I do have one more question, and then Nika, I'll be quiet and let you finish things out. So when people say, would you want their EEG even if it's normal, like even if it's a normal baseline for them, is that still something that's of interest?
Yeah, definitely. So we want to be able to expand our control samples as well. So yeah, if you have data, we will take that too.
Right.
Yeah.
Right. I think a lot of times too, we were talking earlier today that we know that if people go in and put their own data in, and it's more of a parent reported data that usually get the worst case scenarios, because you get parents going, I need help. Let me put this seizure in. Let me put this in. So we want a good mix of data, not just the worst case scenario that could always happen. So I wanted to make sure that was of interest to you too. So that's super valuable. No. Good. So if anybody's watching and you want to be able to give your EEG for research, obviously we're going to be working on getting those uploaded into Ladder. But also if you want to just send that portion, just reach out to one of our team members and we will get that. We'll make sure we get that to Vidya. And thank you. Thank you so much for what you're doing. And Nika, bring us home.
Yeah. Thank you again, Vidya. You are always a treasure to talk to. I love hearing about your work. And so just a reminder to everyone that our goal today for the Big Give is to raise $50,000 to support the Ladder Learning Network. And our board has generously offered to match up to $50,000. So we could have $100,000 to go to the Ladder Learning Network that would support our DUP15Q clinics, that would support DUP15Q database, as well as research and potential future clinical trials. So this money that you would be donating and having doubled really will go far and really will make a true impact on our community. So thank you to everybody who's watched. Thank you for joining us, Vidya. And we will be up next with Ben and Matt, I believe.
That's right. Yep.
Awesome. Thank you so much. Bye. Bye, Carrie.
Thank you.
Thank you. Take care.